What is transcription? The formation of RNA from DNA. To understand that, let's see what's DNA first. The DNA consists of pentose sugar bases. They are deoxyribonucleic. They may be of purines, that is adenine or guanine, and pyrimidines, thymidine or cytosine. Adenine pairs with thymidine with a double bond, and cytosine binds with guanine with triple hydrogen bonds. Phosphate molecule binds the successive purines and pyrimidines in a strand. Thus, a DNA is made up of sugar bases linked to each other by phosphate molecules, and this one strand is paired with a complementary strand. Starting from oxygen, the carbon atoms of the pentose sugar are numbered from 1, 2, 3, 4, 5. The 5' end attaches phosphate and the 3' end attaches the base sugar. Due to this, the DNA folds into a helical structure. Let's see how a RNA is transcribed from this DNA. RNA polymerase acts on promoter region of DNA. The DNA double helix unwinds to form two strands. The strand reading from 5' to 3' end serves as a template for RNA replication. So that is named as the template strand. And the other strand is the coding strand, which is similar to the RNA formed. The coding strand separates away from the template strand. The RNA polymerase moves from one base to another, forming a complementary strand to the template strand. The direction of RNA polymerase movement is always from 5' end to 3' end. Thus, an RNA strand similar to the coding strand and complementary to the template strand of the DNA is formed. So, how does this elongation terminate? A protein Rho factor has ATPase activity and helicase activity. With the ATPase activity, it moves towards the RNA polymerase in the termination site of DNA, and it causes the separation of RNA and DNA by its helicase activity. Sometimes, without the action of the Rho factor, the DNA produces a sequence so that the RNA generates a self-complementary sequence which folds back on itself and it separates away from the DNA. In both cases, after the RNA goes away, the DNA helix zips up back. Thus, an RNA is formed similar to the coding strand and complementary to the template strand. Keep watching my videos. My next video will be on translation. That is how an RNA aids to form a protein. Thank you for watching. Subscribe.